I've suggested. So my title is Natural Product Inhibitors of Poly-ADP-ribose Polymerase as Potential Treatments for COVID-19. Now, what I'm going to do is to, first of all, introduce you to the biology of PARP with regard to physiology and how it contributes to pathology, and then very briefly talk about the current therapeutics that are based on PARP as a target. So when I use the word PARP with high, I mean PARP inhibitor or PARP inhibition, which is not the same as PARP1, which is an isoform of PARP, as you're going to see later. And I suppose it's also important to further emphasize the essence of the emerging model in drug discovery, which is about repurposing. And as we know, that's even going to help us to get more drugs to the clinic faster and at a much reduced cost compared to the traditional model. And then I'm going to be taking some time to review the evidence that is around the repurposing of PARP inhibitors for the treatment of COVID-19. And of course, the centrality of what we're interested in for this conference is the link between whatsoever we say and the natural products, which we are very much interested in. So I'm going to be finishing off by giving you that link and by showing you part of what we have done and part of what some other people have done with regards to that. So in terms of introducing you to PARP, it's a very important enzyme that works for you and myself. As we are seated or whatever posture you are assuming, we know that a lot of DNA damage occurs within us, but we have a system within us that helps to be able to repair that damage. And the protein, PARP, is one of those systems that help us to be able to do that. Of course, it's not just one protein we're talking about here. So there are 17 isoforms of PARP, but the most important of them is PARP1. So I'm going to be focusing on it more. And it's because if you think of the cellular PARP activity, the contribution from PARP1 is about 90%, whereas the other ones will contribute the rest 10%. So what does PARP do? Now, what it does is to use its substrate, which is nicotinamide adenine dinucleotide. So it cleaves NAD, and by so doing, it frees up ADP-ribose. And then it can attach the ADP-ribose to other proteins in a process that is a post-translational protein modification called poly-ADP-ribosylation. Now, it doesn't only attach those units to other proteins, it can use it to modify itself too. There are some of them that will carry just mono ADP-ribose units, and others are going to carry many ADP-ribose units, so they are called poly-ADP-ribose polymerases. So it's involved in genomic stability, as I introduced to you, and most importantly, the repair of single-strand DNA damage. And I'm going to show you later how it does that. And I guess the central pillar, in terms of what we're interested in, is how it is connected to COVID-19. And I will quickly say from here that PARP can be activated by a variety of insults, most notably oxidative stress, nitrosative stress, and inflammation. And of course, you know that these are pathological mechanisms that are very intricately connected to COVID-19. So that tells you a little bit about why PARP is actually important in the context of the disease. So this is to show you the structure of the protein in its uncleaved full-length form. I'm just going to emphasize that to the right-hand side there, you can see the active site. This is where NAD, which is the substrate for the protein, binds. And so we will see later some of the current PARP inhibitors and the way they work in terms of being competitive inhibitors of NAD. So they are competing with NAD for that active site on the PARP protein. So the genetic concept of synthetic lethality is what underpins the use of PARP inhibitors in the clinic. So what happens is when there is a damage to the single strand of the DNA, so PARP protein can facilitate the recruitment of the base excision repair machinery, asking the entire complement to come around to make sure that the repair is is repaired, the damage is repaired, so that it doesn't lead further to double-stranded DNA damage. Now, if you use a PARP inhibitor to prevent the ability of PARP to recruit such repair machinery, that means that those cells cannot repair the single-strand DNA damage that they have incurred. And so when that continues, it can lead to even double-strand DNA damage. And there are different processes, especially homologous recombination, by which double-strand DNA damage is repaired. But that can only happen in competent cells. Unfortunately, for some of the cancer cells, they have an impairment in the HR process, which means that they are not able to properly repair their double-strand DNA damage. And so the reason behind the use of PARP inhibitors, therefore, in such forms of cancers is to prevent the repair of the single-strand DNA damage so that there can be an accumulation of that, which leads to double-strand DNA damage accumulation. And since they're not able to repair that, that leads to genetic instability and the death of those cancer cells. So that tells you that PARP inhibition or the use of PARP inhibitors is part of what we now call targeted therapy in cancers, as opposed to general chemotherapy, which smashes all the cells that come its way. So when you are looking at targeted therapy, you want to look at the molecular signature within a particular type of cancer and use that to be able to selectively target those cancer cells and destroy them without having any effect or significant effect on normal cells. Now, why is it important? Now, we have tumor suppressor genes like BRCA1 and BRCA2, which can be mutated in some cancers. It's a loss of function mutation. And when that happens, it means that these BRCA1 and BRCA2 genes are not able to contribute to the process of homologous recombination, which means that the repair of double-stranded DNA damage is impaired. Now, for you to appreciate the value of having PARP inhibitors in the clinic, if you think of just 12% of women within the general population who are going to develop breast cancer sometime in their lives, compared to women who are carrying either the BRCA1 mutation or the BRCA2 mutation, whose vulnerability increases to a whopping 72% from 12%. So that suggests to you the importance of being able to treat cancers like this. Now, there are some other forms of statistics about the use of PARP inhibitors, but I will just go straight ahead to the other things. And here, I want to show that there are PARP inhibitors now in the clinic. There are 4 of them. They are used as monotherapies. They're administered orally, and they're used to treat cancers with germline mutations in BRCA1 or BRCA2. And these can be gynecological or non-gynecological cancers. Now, PARP inhibitors are also now being proposed for use in combination chemotherapy, but that has not reached the clinic yet. So as you can see there, from 2014 to 2019, 4 PARP inhibitors were approved for clinical utility by the FDA and the EMA, and those are their structures. Now, I want you to look at those structures and then look at what we have here in terms of NAD, which I told you that it's the substrate for PARP. So in the center there on top is NAD, and the red part is the nicotinamide moiety of NAD. So what PARP does is to cleave the N-glycosidic bond here to free the ADP-ribose and then attach it to other proteins. So if you think of the structure of NAD and the structures of the different PARP inhibitors, some of them in clinical trials that have been produced, you can see in red what is common, the common moiety there. And I told you that is just illustrating that they are all competitive inhibitors of nicotinamide. And if you look to the left-hand side, I have illustrated two of those clinically used PARP inhibitors showing you the same nicotinamide moiety that they carry. And most of the efforts in drug development would be to increase the potency of the different drugs or their selectivity. So they are used to treat cancers, and that's enough. What do we want to do now? It's been shown that PARP inhibitors can be repurposed for a lot of other indications, most importantly, neurological and neurodegenerative diseases, which we have worked upon for many years now. And this slide illustrates to you what happens when a glutamate receptor is overstimulated, and it eventually leads to DNA damage that sensitizes PARP, and PARP, through a series of downstream mechanisms, is going to lead to DNA fragmentation, nuclear condensation, and eventually death. What's also important to note is the fact that PARP activation consumes NAD, and we know that NAD gets synthesized from ATP. So when there is sustained activation or overactivation of PARP, then eventually that's going to lead to a depletion of NAD content within the cell. And as a consequence, that's going to lead to a depletion of ATP within the cellular pool. So what is it about PARP that makes us to want to think of its potential to be used even in the treatment of COVID-19? Now, PARP is anti-inflammatory, it modulates cell death, and it can also participate in cytokine storm. I'm not going to say much about a cytokine storm because this afternoon we have an excellent presenter in Dr. Olajide, who is an expert in inflammation, and he's going to be telling you a lot about that. But what I want to emphasize here is that oxidative or nitrosative stress is important to inflammation, and each of them can cause DNA strand break, and remember that profoundly activates PARP. And either oxidative or nitrosative stress is relevant to inflammatory tissue damage. And as we see in acute respiratory distress syndrome, which is one of the features of the cytokine storm, when the body produces significantly elevated levels of circulating cytokines, and it begins to fight itself, and that can even lead to organ damage. So it's been shown that PARP inhibition can limit inflammation-induced tissue damage in various animal models. And also, and in particular with reference to SARS-CoV-2, it's known that reactive species production actually increases in COVID-19, which suggests that there is PARP activation and there is PARP-induced cell death. And so it's been reported that SARS-CoV-2 infection actually induces PARP activity, which tells you of the connection to the use of inhibitors of this enzyme to be able to treat COVID-19. And even in similar viruses that are called positive sense single-strand RNA viruses like Zika virus, PARP inhibition has actually been shown to reduce cell death. We also know that PARP is involved in regulating innate and adaptive immunity, and it can even confer protection on the lungs in cases of acute lung injury. We know through preclinical models and also in some instances in humans that PARP inhibition is protective, whether in the context of ARDS or in the context of COPD. So these are various lines of evidence that connect PARP with the pathophysiology and the symptomatology of COVID-19. We also know with regard to the different cytokines that are involved in the cytokine storm, most importantly, IL-6, IL-1, and probably TNF-alpha. So in that context, we know that PARP inhibitors can actually reduce the expressions in different organs of IL-6 or C-reactive protein or IL-1 beta or even TNF-alpha levels. And so we argue that the use of PARP inhibitors is very important in the treatment of COVID-19 and should be trialed. Now, this figure just shows you the vicious cycle that occurs when PARP is activated in those different models of inflammation, whether connected to COVID-19 or to other pathologies that include inflammation. So if you have excessive levels of oxidative or nitrosative stress, they can actually induce DNA damage and thus activate PARP, which goes ahead to induce cell death through pathanatos and can also deplete the cellular levels of NAD and ATP also as a consequence. And in that process, you can have necrosis, which induces inflammation, including the release of NF-kappa B and inflammatory cytokines. And so that very vicious cycle continues. So this is just to show you some efforts to think of some of the inhibitors of PARP as potential treatments for COVID-19. And I have nicotinamide on top here, which I showed you when I was describing how PARP cleaves the glycosidic bond of the NAD. And so, in fact, some people have been quite bold to say that PARP activation is probably the terminal point in a sequence of events that lead to patient mortality. And they argue that it should be the focus of COVID-19 immunotherapy. And there is also a connection between the regulation of the PARP gene and the aryl hydrocarbon receptor that is used by many toxins. And we can find the overexpression of some of these in COVID-19. We know that coronaviruses can elicit AHR activation. And so some people have actually proposed that nicotinamide could be one of the drugs known that could be trialed for use in COVID-19. It's a low potency PARP inhibitors, but
minutes, Amos.
Yeah, but nevertheless, it can be used for the treatment of COVID-19. And then other people have suggested compounds which are also PARP inhibitors like 218, CBL218, which is connected to different mechanisms, including inflammation, the nucleocapsid protein of the COVID-19, the virus, and a number of other mechanisms. And some of these PARP inhibitors that have now been tried for COVID-19 can work either alone or in combination with other drugs that are being trialed for COVID-19 treatment. This is to show you another one, stenoparib, which actually increases, that brings about dose-dependent inhibition of SARS-CoV-2 without actually eliciting toxicity. So there is a little bit of a conundrum between what happens to PARP 1, 2, and 3 versus the smaller PARPs, but we know that most of the PARP inhibitors will target PARP 1 to 3 and not really the other PARPs which might have their own antiviral effects. We have shown before that 2 compounds that we got from a screen of 5,000 compounds are PARP inhibitors, and we have shown the structure activity relationship indicating that it's obligatory to have the methoxy at position 4', but if you have it an additional methoxy at 3', you don't compromise activity, but you can actually improve metabolic stability. Others have also shown the potential of epigallocatechin gallate and theaflavin in inhibiting PARP. And as you see here, the red line is about the cells that are having the mutated form of BRCA2, which are more sensitive to these drugs compared to the ones that are carrying the wild type form. So in conclusion, we have significant evidence that PARP1 can actually, PARP1 or PARP inhibition could be amenable to repurposing for treating COVID-19. So we are suggesting that clinical trials should be undertaken to see whether existing clinical PARP inhibitors could be so used. And it's going to be very beneficial because we reckon that if you do that, you wouldn't even have to use very high doses of these compounds as you use in the treatment of oncological indications. And so it's worth trying. And finally, natural products with PARP activity, just as I've shown you, and as people are still working on them, could be potential therapeutics for COVID-19. So there is a vast world of opportunity for using natural products to be able to treat COVID-19. I want to thank my PhD student who has done some work along this line. I could not show the data because of limited time, Yomi Benson, and then some of the people I had worked with, including at Johns Hopkins in the States some few years ago. And I want to thank all the members of the CNPD for their collaborations, for their support and different ways that we have been working together. I would like to thank the audience for listening. Thank you very much.
Thank you, Amos. Very clear presentation. Thank you. Okay, can I just remind people, if they've got any questions for Amos and the other presenters, if they could put them in the chat, and I'll try and keep my eye on them.